Everything is fun in hybrid land. So it's really great to welcome Catera to the QBI community. Catera is one of our newest assistant professors on campus. She's a Chan Zuckerberg Biohub investigator and assistant professor in the Department of Bioengineering and Therapeutic Sciences. Still can't hear me? Or
They can, just can't see me.
you can't see me. We're going to need to end the slideshow. It won't work. It's not a thing.
Oh.
Yeah, because of the laser pointer aspect. So there we go. Okay. There we go. And then now is everything working? Yeah. You can see us? Okay, great. So Professor Wilder got her Ph.D. At Georgia Tech, where she did really cool work on proteases and various cancer models, and then moved over for her postdoc work to the West Coast, the best coast, where she worked in the Hoffmann lab, really integrating modeling and systems biology studies, bringing this engineer's perspective to the immune system. We're really excited to have her here at UCSF, where I think she's working at a very fruitful interface between engineering, modeling, and immunology. And I'm really excited to learn a lot today about interferon signaling. So, Professor, the floor is yours.
Thank you, Jamie, and apologize for all of the technical issues. I'm excited to share my work today, though, that I did as a postdoc. Give me a second while I get this pointer. That I did as a postdoc, like Jamie said, at UCLA in the Hoffmann lab, and kind of at the end, if there's time, we're running a little late, so I might not have as much time, but if I have time, kind of like what I want to do, since I'm here, I've been here only a few months, so still trying to get things up and running. So the immune system is pretty complex, but most people kind of think of it as the innate immunity as well as the adaptive immunity. So in this schematic, you'll see epithelial cells, and just for context, usually I think of infections of the lung. So we're going to assume that these are lung epithelial cells that are infected with a virus. And again, you have this adaptive immunity where you have these macrophages and dendritic cells that will come to the site of infection. And then later on, you'll have the induction of your, or did I say that wrong? Your innate immunity with the macrophages and dendritic cells. And then later on, you have your adaptive immunity with the T cells and B cells for long-term memory. But I'm really interested in kind of the innate immunity of these lung epithelial cells in particular, and how they are contributing to the control of infections. And so some of the reasons why I'm interested in lung epithelial cell immunity is because lung epithelial cells are the primary targets of infection, specifically when you think about lung respiratory infections. And also the immune system or the immune response is not linear. So you do have, again, people think of innate and then adaptive, but there's going to be some feedback between cell types throughout this whole process, so that the immune cells can be informed. And that's going to be primarily, again, through some of the things that are happening within these epithelial cells. And epithelial cells themselves can secrete factors that are important for immunity. So they secrete some of these interferon cytokines. And interferons are important for putting these cells into this antiviral state where they can try to restrict the replication of the viral replication. And the importance of these interferons can be seen down here in these Kaplan-Meier plots, where mice were infected with influenza A. And we have wild type mice as well as, sorry for the small print, but you have various receptor knockouts for these interferon cytokines. And so right now we won't get into the different types of interferon receptors, but in general, you see that without the signaling of these interferons, the survival of these mice days post-infection is decreased compared to the wild type. So these interferons are definitely important for just overall immune health and survival. So to go into a little bit of signaling, because I was really interested in what's happening intracellularly with these interferon responses, is when you have viruses, you will have viral DNA or RNA, which will activate some of these pathogen recognition signaling pathways, which induce activation of some of the IRF transcription factors, as well as NF-kappa B. Those will bind to the promoters of these interferon genes, which upon induction, you'll get secretion of these interferons. And so this is a secondary response. And so this secondary response will then activate this transcription factor ISGF3, which will then translocate to the nucleus and activate important antiviral genes. Some of them are listed right here to the right, as well as other regulatory factors as well. And so again, these interferons are kind of the secondary response after there's a recognition of these pathogens. And one of the things that I was really interested in as I started my postdoc was, why do you have so many interferons that can activate some of these antiviral responses? And so here we're looking at type 3 interferons as well as type 1. And you can see within the different types, you have a diverse amount of interferons. Some of them are cell type specific or species specific. And so I wasn't really interested in terms of within a family, but I was interested in across family. So type 1 and type 3, how can you activate different receptors, but then get a different response, even though you're activating the same transcription factor, again, which I mentioned before is ISGF3. And so well, I guess one question was, can you get different responses, although you're activating the same signaling network? And if you can, how is this happening? And so there were some studies out already that kind of showed that indeed you can get differential responses with these type 1 versus type 3 cytokines. And I won't go into a lot of detail, but I'll kind of explain this first plot up here. These were mice that were infected with, again, influenza, but then treated with either interferon alpha, which is a type 1, and that's seen in the circles, or the type 3 interferon lambda, which is in the triangles. And you see that this circles, which again is interferon alpha, it induces IL-6 protein. So this is IL-6 protein that was taken from a BLA, a bronchoalveolar lavage. And so you see there's increased inflammatory cytokines. Again, they measured IL-6, IP10, which is CXCL10, as well as MCP1, which is CCL2. So just two important cytokines and chemokines that are known to be involved in inflammation. And these type 1 interferons differentially induce that compared to the type 3, which again, you see, or you can't really see because they're behind the vehicle control, but they're in the triangles. And so even when you look at specific cell types, so they measured the same cytokines, but in macrophages and DCs. And again, in the circles, you see there's an induction of IL-6 across the board for all of the cytokines that the type 3 interferons do not induce. And so it does seem as though, at least in these cell types, that there is a type 1 specific induction. But it wasn't.
How do you know that the lambda ones are even working at all?
So that's the question. Precisely.
Okay.
Is it just that those cell types didn't have the receptors for those cytokines, or did they, and then they just weren't responding? And to your point, indeed, macrophages actually don't have, and dendritic cells don't really have receptors. And so this is another study that measured gene expression of some of this top row are the receptors for either the type 3 interferon. These first two, IL-10 receptor beta and interferon lambda receptor, are the subunits for the type 3 cytokine, and IFNR1 is for type 1. And you do see, so UA or AM is the alveolar macrophages, and you see they don't have expression of the receptors for the type 3 interferons. But you do see these epithelial cells, which you see in EC, do express both of the subunits needed to bind and activate the signaling pathway for type 3 interferons. And even though it's maybe not as important, they did have mice that were either control mice or treated with AIV, so again, influenza virus, and you do see that there's increased expression of these receptor upon induction. And so I was interested again in looking at lung epithelial cells specifically because they have receptors for both the type 1 and type 3. And in that system, can we get different responses, although we're activating the same transcription factor activity or transcription factor pathway? And so I used MLE lung epithelial cells, which is a murine epithelial cell line, and I treated them with either interferon beta or interferon lambda, which beta is in blue and lambda is in red. And I did mRNA-seq sequencing, and we're looking at global gene expression in a PCA plot. And each dot is representative time, so it's a time course experiment where smallest dots are less time, and then we went up to 36 hours. And so one of the things that I think you can clearly see is there's just different gene expression patterns, especially in the middle of the time course, where these dots over here that are gene expression induced by interferon beta is at 4 hours particularly, are very far from the 4-hour time points induced by interferon lambda. And then even when you look at the number of genes induced between both of the cytokines, again, around this kind of 4-hour mark, you see that there's this large amount of genes induced by interferon beta, close to 400 genes, that is not induced by this interferon lambda. And so you see this temporal pattern that I'll kind of get into a little later, but it does seem like so there's gene expression differences as well as temporal differences. And so even though before I was looking at global gene expression, we wanted to also see in genes that are similarly induced. So if you have a particular gene that's induced both by beta and lambda, do they induce the expression at a similar fold change? And so we are plotting induction of genes by beta, and that's x-axis, and induction by lambda is the y-axis. And you do see that at 2 time points, I'm looking at 4 hours as well as 24 hours is in gray. 4 hours is in black. That even the genes that are induced by both, there is a preferential induction by beta at the early time points and then by lambda at these kind of later time points. And so this kind of led us to want to look at each individual gene using this heat map. And we were able to cluster the genes into 2 different clusters. One was genes that are induced by both beta and lambda, which is this bottom cluster, as well as genes that were only induced by beta, which was this top cluster. And so that in and of itself was something that, and this was grouped based on p-values, so p-value of greater than 0.05, either with both stimuli or either just with beta. And so it was interesting to see that there is a subset of genes that are preferentially induced by interferon beta that we're not seeing with lambda, which again is this top cluster. Another kind of takeaway from this was that even with the genes that were induced by both, similar to what we saw in the previous slide, there is this temporal pattern that with interferon beta, you have this transient response that peaks at around 4 and 6 hours. But then with with lambda, you have this delayed induction that peaks at these later time points around 24 and 36 hours. So we were really interested to see what are these genes that are induced specifically by interferon beta? Are there functional relevancies to immunity? And so this cluster 1 gene, you see this is gene ontology analysis, and we looked at the different functions of the genes within cluster 1 as well as cluster 2, which is seen at the bottom. And one of the biggest takeaways was these cluster 1 genes had functions that included cytokine production and inflammatory and adaptive immune response that was not seen in the genes that were in cluster 2. And so it does seem as though the genes, or at least some of the genes in cluster 1 do contain functions that could lead to pro-inflammatory responses. And so even though this is interesting that these cytokines, although they're activating the same transcription factor, can lead to differential responses, we were really curious to figure out what is the mechanism and what's causing this preferential induction of the transcription factor and, again, these gene expression responses. And so one potential reason could be differences in binding affinities, because, again, these cytokines have different receptors. And the interferon beta receptor, these are just binding affinities for the cytokine to the receptor. You see that it has a nanomolar, 100 and 0.1 nanomolar KDs, while the interferon lambda has higher ones that are more in the micromolar range. And so there are differences in the binding affinities. So we just looked into the literature to see if other people had kind of looked at the importance, and indeed people had. And so one way to kind of address this difference in binding affinity is just by adding more receptors on the surface of these cells for either of the receptors, either the type 1 or type 3, and see if that changes either the gene expression patterns or magnitudes. And you do see that in wild type cells, which is this first column, when you overexpress or when you don't have, when you just have the wild type receptors, you have particular kinetics that are maintained even when you overexpress the receptor. So the second column are the same cells, but overexpressing the IFNR receptor. And the same thing is done with the interferon lambda receptor in red, where, again, these are wild type cells treated with interferon lambda and measuring gene expression. And then when you overexpress the interferon lambda receptor in the last column, you see that it maintains the temporal kinetics of these genes that are induced. And so it does seem as though the binding affinities does not play a role in the temporal kinetics of the genes that are activated. And so that kind of led us to think that maybe some of the intracellular pathways are important for regulating some of the differences that we're seeing between the type 1 and type 3. And so some of those things include feedback loops that are formed. So again, upon binding of the ligand to the receptor, you'll have activation of the transcription factor ISGF3, which is seen here. But then you'll have these positive and negative feedback loops. So STAT3 or STAT1 or STAT2 would be a positive regulator, as well as some of these negative regulators. So I was really curious to see how are these kind of feedback loops playing a part in terms of regulating the system. And also one of the hypotheses was that there's a temporal code. So maybe even though you're activating two different receptors that are binding the same transcription factor, that there's some temporal code of the transcription factor that is inducing particular subsets of genes over others. And so one of the first things to do was just to measure the transcription factor activity itself. And so this was done by using mobility shift assay. So this is an assay where we took an oligo that contained the binding motif for ISGF3. And so wherever you see a band, it's actually the trimer itself, the STAT1, STAT2, IRF9 trimer that's bound to the oligo that's tagged with the radio label, a P32 radio labeled oligo. And so you're actually looking at the transcription factor of interferon beta induced cells or interferon lambda induced cells over time. So we did up to 12 hours. It's also quantified over here to the right. And it's pretty clear that with interferon beta, the transcription factor activity is potentiated during starting around 1 hour, and it lasts until about 6 hours, 4 to 6 hours. It starts going down around 4 hours. And that's in contrast to the type 3 interferons, which doesn't actually induce this potentiated phase, but you actually see this trough, this down regulation around 1 hour that then kind of is recovered in the later time points. And so this was really intriguing to us, one, because of the timing. So the timing kind of indicates that this potentiated phase could control some of the gene expression that we saw with interferon beta, and also just how is this happening? So one of the first questions that we wanted to see was, are the differences that we see in the dynamics of the transcription factor important for gene expression? And can it cause differential gene expression like we saw with the RNA-seq data? And so one of the ways we did this was by using a ODE mathematical model to simulate RNA abundance. So the biggest takeaway is that this model is just looking at or simulating RNA abundance as a function of transcription factor binding and RNA degradation, and looking at RNA abundance over time. And so I won't go into the details of the model, but can talk about it if needed at the end. But we fit the data or fit the model to our RNA-seq data. So the model fit is the line, but the marks are the data. And so what we did was we just took that RNA-seq data at each time point, and we took the median, our median gene expression for each time point, and also for each cluster, and then calculated that, and then used that as the data to fit the models. And so with that data, we were able to get models that did a great job of actually recapitulating the temporal dynamics that of both the data. So we fit the model to both interferon beta as well as interferon lambda. And so that kind of, sorry, there's noise, but, and so that was really, the fact that we were able to fit both data sets using the same model did indicate, and I should re-emphasize that the only thing changing in this model were the temporal dynamics of this ISGF3. And so the fact that we were able to fit both the beta and lambda data sets does indicate that only by changing the temporal dynamics of the transcription factor binding, we were able to recapitulate the differences that we see in the gene expression responses. And so the temporal dynamics can play a part in regulating these gene expression dynamics. We were able to see that not only for the genes that were in cluster 1, which again included some of these inflammatory genes, but also we were also able to see that for the genes that were in cluster 2, which had a different temporal pattern. And one of the things that was interesting that I won't get into at this point, just for time's sake, but could talk about it later if people were interested, was that we did have to have 2 different models to fit the genes in cluster 1 as well as cluster 2. And so while the temporal dynamics of the transcription factor do play an important role in controlling gene expression, it does seem as though there are different regulatory mechanisms on top of the temporal dynamics that can control both of the genes within cluster 1 as well as cluster 2. And again, I won't get into that, but most of the data is published in bioRxiv, and it's also under review. So some of that, if you're interested, you could go look at that manuscript on bioRxiv. But for this talk, I wanted to get into like the mechanisms of what's controlling the temporal dynamics, and a model that we use to also kind of answer that question of, we know that this temporal dynamics are important, but what are the mechanisms? And so we used another ordinary differential equation model, but this time not just of RNA abundance, but of the actual signaling pathway. So it simulates when you have a receptor binding to a ligand binding to a receptor, a lot of the pathway regulators, and then the output is transcription factor activity. So ISGF3 activity or abundance, I guess, in this case. And so it's a model, it has 41 species and 75 reactions. And the data that we used to fit this model included a bunch of time point experiments of different Westerns, IMSAs, as well as qPCR data. Again, this is some of the data that we used to fit, and the most optimal the most optimal model that we were able to find was this model that's in this dark blue line. And you see that for these different species, we were able to get pretty good fits. And we did have to change 21 parameters compared. So this was a previously published model, and we had to change 21 parameters to to actually have the model fit to our data. There were some differences between the experimental pipelines that the previously published model used, but we were pretty excited that we only had to change a minimal number of 21 parameters to get this model to fit to our data pretty well. And we also saw that for, again, all of these other species. I won't, again, go into the details, but the takeaway was that we were able to have a pretty robust model that can look at transcription factor activity within our experimental system. So now that we had a model, we wanted to test the model to see, can it predict anything about the transcription factor responses in our experimental system. And so we thought that maybe the dose was important for this potentiated phase of ISGF3, which again, we saw was important for these pro-inflammatory genes that were induced in these lung epithelial cells. And so with the model, we were able to see that by decreasing the dose of interferon beta, you were able to still get the activation phase, but just decrease this potentiated phase, especially at some of these like 0.5 and 0.25 x of the cytokine. And then also when you look at just the 1 hour and 4 hour time point with the model, you again see that there does seem to be this dose dependence of the potentiated phase where you're still able to get activation at 1 hour at these higher doses, but that this potentiated phase that occurs at 4 hours is just more dose dependent. We also saw that the potentiated phase that we saw with ISGF3 is sensitive, at least the model predicts that it's sensitive to duration of stimulation. And when you only have about 15 minutes of stimulation in this pulse condition, which is in purple, you no longer get this potentiated ISGF3 transcription factor activity. And so these were all predictions that the model gave, that this potentiated phase is dose dependent as well as duration dependent. And so we tested that hypothesis with our experimental system, and we indeed saw the same thing where the dose did play a part in having this potentiated phase. And even at lower doses, like 1 units per mil, you were still able to get activation within the first hour. And you did get a sustained response, but you just didn't get this potentiation of ISGF3. And again, you see that with the bar plots where at 1 hour and 4 hour, where you get this decrease in this potentiated phase at 4 hours with the lower doses. And similarly, with the duration, we saw that when you had only 15-minute stimulation of the cytokine, you got this induction at the initial activation phase, but that it was quickly downregulated. And so this potentiation, this potentiated phase seems to be important for, or it seems to be dose and duration dependent. And so we wanted to see how would that impact the gene expression responses, particularly of the inflammatory genes. And so we did similar mRNA-seq experiments where we measured the global gene expression. And so what I'm showing now is the same low condition in the light blue, as well as the pulse condition of 15 minutes of stimulation in the purple. And you see that those dots now more closely match interferon lambda, which didn't have the potentiation as well, compared to, again, at this 4 hour time point with the high dose of interferon beta, you had this gene expression that occurred out here in the PCA plot. And even when we look at the heat maps, you see that these gene expression profiles for the genes in cluster 1 are also diminished when, again, you have this low and pulse condition. And when you look at individual genes, again, you see the same thing where interferon beta, this is gene expression. This is the mRNA-seq data, but just for the individual genes. So for some genes like IL-6, TNF-SF10 and CCRL2, which are important for pro-inflammatory responses, you see induction with the high amounts of interferon beta, but with lambda or with the low amounts, you no longer see induction. And so I think we were excited to see that the temporal dynamics of the interferons played an important role in controlling these pro-inflammatory genes, but we still weren't sure what was causing this potentiated phase. And so we decided to see if I mentioned earlier that there's positive and negative feedback loops that occur within the system. So we wanted to see, were these some of the regulators that cause the potentiated phase of interferon beta stimulation? And so first we just stimulated these cells with cyclohexamide on top of the interferon beta. And we did see that this is, again, the ISGF3 transcription factor binding that was quantified from some of the mobility shift assays. And you see that the potentiated phase, when you block protein synthesis, does not occur. So it does kind of infer or suggest that that there is a feedback loop that's important to cause this potentiated phase. I also just want to note that the activation phase still occurred, so you're still getting activation of the transcription factor. It's just this secondary response. And so we decided to look at some genes that could be, that were known to be positive regulators of ISGF3, including STAT1, STAT2, and IRF9. And so the time, and this is qPCR data for each of these transcripts. And so the timing of IRF9 in particular, we thought was interesting, where at 1 hour you had induction of IRF9, which would kind of correspond with the timing of our potentiated phase. And even when you look at the protein amounts of the nuclear, so this is nuclear Western blots that's been quantified. And so the nuclear IRF9 also increases during, or pretty early on during the time course, where even at 100 minutes, you have this large fold increase of IRF9 present within the nucleus. And so we were, and I should say the model actually predicted that IRF9 feedback as well as STAT2 feedback could play a part of reducing the potentiated phase that we saw with ISGF3. So again, this is the model simulation. The dots are the data. The blue curve is the previous model simulation that you saw with the potentiated phase. Yellow is where we basically block the STAT2 feedback loop, and then purple was where we block the IRF9 feedback loop. And so you do see that you don't get this potentiated phase. It's reduced for both of those. So since kind of the timing of the gene expression, as well as the translocation of the protein and the model kind of suggested that these feedback loops could be important, we decided to test that experimentally. And so I took these cells and I knocked out the transcription factor promoter site, or not the whole promoter, but I used CRISPR-Cas9 to mutate the transcription factor binding motif so that you had reduction of transcription factor binding at the STAT2 and IRF9 gene loci. And so you're not actually, it's not a complete knockout of these genes. It's just trying to reduce the feedback loops. And you see that in this data. So on the left is the STAT2 mRNA as well as IRF9 mRNA. And so you see at the unstimulated time points, you still have the same amount of STAT2 and IRF9 in the system, but then at 1 hour post-stimulation, you do have this reduction of STAT2 in the yellow as well as IRF9 in the purple, which again indicates that it's more of a feedback loop mutation and not a knockout. And so within these cells, we then decided to measure ISGF3 activity. And you do see that indeed, when you block these positive feedback loops, you do get a reduction of this potentiated phase of ISGF3. And when we looked at gene expression of some of the pro-inflammatory genes that we had seen before, you also get a reduction in the gene expression of these particular genes, indicating that these feedback loops could be playing an important part in regulating pro-inflammatory conditions in these lung epithelial cells that's induced specifically by these interferons. So kind of in conclusion of what I did as a postdoc, These type 1 interferons seem to be important for regulating inflammatory genes. And so I do just want to highlight the importance of that a little bit more, because traditionally these type 1 interferons are known to be antiviral. They're thought to have these cell intrinsic responses where just it controls the cell itself. But this is showing that not only are you having these cell intrinsic antiviral responses, but you can also activate cell extrinsic pro-inflammatory responses in these lung epithelial cells by this cytokine that's not traditionally thought to be pro-inflammatory. I also showed that this is due to a temporal code where certain types of interferons or certain types of interferons, so type 1 interferons has a temporal code that has this potentiated phase that can cause the induction of these pro-inflammatory genes, and that type 3 interferons has a separate or different temporal code that lacks the ability to activate some of these genes, although it still acts an antiviral response. So these type 3 interferons, it still has this cell intrinsic antiviral response, but is not activating this more cell extrinsic pro-inflammatory. And again, that's due to this temporal code. And so this is important for understanding the mechanisms of inflammation, especially again, since these type 1 interferons are not traditionally known to be pro-inflammatory regulators, and also just indicating that dynamics are important. So I think a lot of studies sometimes will only measure 1 or 2 time points, but it's really important to understand what's happening throughout a response to really figure out regulators that are important. So that's what I did as a postdoc. And so for the last few minutes, I'll just kind of describe kind of what I want to do next and ways that people could collaborate or work together. And so again, these type 1 interferons, I think, are beginning to become more and more appreciated for their pro-inflammatory roles, but their actual kind of, if it's a direct cause or indirect cause is still not clear, at least in vivo. And so that's kind of shown down here at this bottom plot. So I guess I'll just say briefly, at least with SARS-CoV-2, one of the hypotheses was that a lack of induction of these interferons played a role in long-term disease severity. But even down here, when you look at, this is patient data of patients with COVID-19, and pink the patients were basically grouped as moderate or severe. And so pink was severe, and blue was people who had moderate COVID-19. And so these plots to the left are the actual individual patients, and they measured the amount of either interferon alpha or lambda in the blood of these patients. And these inserts are basically regressions of this data. So that's why I'm just going to focus on the regressions. And I should say that this was looking at the amount of these cytokines in the blood at different days post symptom onset. And so they did show that in patients that had more severe COVID-19, their both interferon alpha and lambda increased over time. And that was compared to the patients who were indicated as moderate, which again was in blue, which some of them started off with higher amounts, but then decreased or was more stable over time. And so it's not clear whether interferons are directly causing some of the disease progression or inflammatory responses that are occurring throughout a disease, or if just lack of an interferon response early on during the response is what's causing some of these cytokine storms or pro-inflammatory conditions. And so, so the direct or indirect role of these interferons is not clear. And also these interferons, they have stochastic gene expression, and so that can impact how these responses are occurring in vivo. And so this plot on the left is data. These are actually dendritic cells that are infected with, I forget the name, with the virus, Newcastle disease virus. And they looked at the amount of viral mRNA compared to the amount of interferon beta mRNA. And this is just a correlation. And you can see that the actual amounts of viral mRNA to And this is single cell, actually, this isn't single cell sequencing. This is in situ hybridization, but it's looking at the amount of mRNA of both the viral mRNA and the interferon beta in a single cell. And they show that it's not correlated. So even though you have some cells that are infected with the virus, they're not actually inducing high amounts of interferon beta. And you also have some cells that have high amounts of interferon beta that actually don't have high amounts of the viral mRNA. And so the interferon beta response can be very stochastic and random. That's also kind of seen here in this data as well. This is single cell RNA sequencing. This top plot is also looking at the RNA viral mRNA expression. And this is interferon beta as well as interferon lambda gene expression in these single cells. And again, you see that there isn't really a correlation with viral infection and these interferons. And I'm really interested in trying to understand how that plays out spatially, especially because concentration gradients can form within a 3D space. And so do you have some cells that are high interferon beta producers that around those cells would be causing more inflammatory responses? So that's kind of depicted in this cartoon where you have some cells that have these high amounts of interferon beta, which could cause more inflammation, and is there clusters that could be seen spatially? And so this role of distance from cell source, I think, will be really important. Additionally, I think the kind of state of the matrix could be important. If you have like a normal versus diseased tissue, you could have differences in how the cytokine can propagate or diffuse through the tissue. And is that different for either the interferon beta as well as interferon lambda? So for interferon beta, is it able to be transported through the tissue more readily than or less readily than interferon lambda? Are there receptors, differences in receptor expression? Does that cause differences in how the cytokines are able to diffuse in tissue? And then again, do these differences in the cell state as well as receptor abundance cause clusters of cytokines that lead to higher concentrations, which I've shown the concentrations are very important for kind of these pro-inflammatory gene induction. So I'm going to do this a number of ways. I won't go into details just so I can leave some time for questions. But I do want to do some mechanistic work as well as using some computational tools to be able to predict some of these responses that are happening spatially. And then lastly, I'm hoping to leverage the information that we gain from this to have more engineered immuno-based systems where we can control some of these interferon secretions in dendritic cells as more of a controlled therapy. And so I'll take any questions and thank you guys for listening. Yeah.
We understand this IFNNS that sort of seems to be such a key input to the model. So you're cracking open cells at various points and then purifying the transcription factor and running the IFNNS, or it's done in assay, or how does that work?
Yeah, so for those on Zoom, the question is basically how is the IFNNS, the assay, how is it performed? And so I lyse the cells, I get a cytoplasmic as well as nuclear extract. And so since I'm only interested in the active forms, I take the nuclear extract and basically add this oligo again. So the oligo is
It's just you do an ultracentrifugation of the lysate, you take the nuclear portion.
And then we just add the oligo to it that's radio labeled. And so if there's transcription factors that are actually in complex, they will bind to the oligo since it has the binding motif. And so we do have controls to make sure that this is like an actual signal and that it's specific. And so, but it's pretty simple, straightforward, except for that I'm not purifying.
And then what are the dynamics of the sort of the cytoplasmic nuclear shuffling transcription factor? Like that? Is that incorporated into the model?
So we did, since we had the cytoplasmic fraction, I also measured phosphorylated forms of the protein. I didn't do IMSAs on those, but I measured the the subunits. And so we do see that the kinetics are similar. So you do have this activation. I don't know how much of that is because of shuttling. Like, I don't know if it's just primarily, the thought is they're primarily in the cytoplasm initially, and then they're just translocated to the nucleus. And then once they become inactive, they will then be shuttled back out. But I don't know, I don't have rates that I could give you that kind of indicate how fast that's happening.
Curious what this kind of temporal pattern or either one of these interferons play in autoimmune diseases or maybe like a long COVID, if they're known to be off.
So the type 1 interferons specifically are definitely known to be regulators of lupus, which is one autoimmune disease. I will say that I guess my experimental platform or setup is definitely looking at more transient responses, because the latest I went out was 36 hours for gene expression and then 12 hours for the transcription factor activity. I think there's definitely, at least in vivo, and when you have multiple cell types, they can also secrete interferons, and macrophages, and particularly, and I think DCs are known to have these like positive feedback loops where if you stimulate them with interferons, they will also secrete interferons. So I do think that kind of this ongoing interferon induction, at least in tissue or in vivo, could be playing a part in some of these pro-inflammatory conditions. In terms of autoimmune, that could also be happening in like an autoimmune setting as well. But I think, I don't know particularly of what would sustain that over like months and years. I think differences in genetics is probably one factor that could be playing a part, but it wouldn't just be stimulus induced, I think, in those contexts.
Sort of the virtue that when you start going to these three-dimensional models and you start incorporating the feedback between cells, what time scale do you think you're going to be needing to, or able to model out to? And how, you know, it seems like it might, the ODE approach will get carried pretty quickly. Incorporating the spatial aspect. Right?
Yeah. So
how does number 2 do it?
Yeah, so I'm hoping to at least model out to like days, a few days. I'm not sure if we could go longer than that because of the timescale differences that you're talking about. For the modeling aspect, I was also going to do some machine learning. So it won't be as mechanistic. We won't understand like how it's happening, but at least we could maybe have correlations of spatial patterns to immune cell, like amount of immune cell recruitment or lung pathology. And so we might have to just do some things that it's like, it's correlated. So we could use this like as a diagnostic, but the ODE model, I think I would need to work with a little bit to see like how far we could go to have some predictions, even though I do think it would be important for longer term, like weeks or months. It would be pretty cool to have that.
We have a question.
Sure. Rich Loxley. Hey, Rich. Says, great talk, Catera. In the first hour, I missed why type 3 interferons don't stimulate transcription at the positive feedback loops, like STAT and IFCs. Is there some other factor at play?
So most of the work that I presented was focused on how this potentiation is occurring with the type 1 interferons. And I didn't show the data about the type 3s in terms of how they're regulating the system. One, because I'm still trying to tease out how it's happening. So I didn't show this, but type 3 interferons, they can also induce this positive feedback loop in terms of induction of STAT2 and IRF9. So they do induce the STAT2 and IRF9 transcripts, but it seems as though they inactivate their kinases, which prevents the complete positive feedback loop to occur. And so I think I'm still trying to do additional experiments to understand like what's happening at the receptor proximal level to understand how are these receptors downregulated and why can't they activate the induction of some of these STAT2 and IF9 transcripts within the first hour of stimulation.
All right. Well, we're at the hour. So thank you, Zoom people. Good to see you, Rich, online. I've been hoping you two would meet at some point. So hopefully this catalyzes that. Thanks, QBI folks, and let's thank Catera.
Yes, thank you.